I've drawn some pictures as well, but this is also probably a better one. We know how I draw. The blue one is the enzyme part. This is the protein that was encoded in the DNA and got transcribed and translated into a protein and made, and this is the enzyme there. This particular enzyme is shown needing a coenzyme to complete that active site. You can see how that fits in. It has to complete the active site and is necessary in order to make a functional active site. This particular enzyme also needs an inorganic cofactor. You can see how that metal ion fits in here as well. And all of this together is going to be a fully functional enzyme, an enzyme that can do its job. So a little bit of vocabulary. When you talk about an enzyme that is made but is not fully functional without cofactors, added cofactors, that particular portion of the enzyme that is made is called the apoenzyme. So that's the protein that gets made, the coenzyme or the organic cofactor, and then the inorganic cofactor as well. Remember, if an enzyme needs a cofactor, it will always need that cofactor to function. Some don't need any cofactors. Some only need an inorganic cofactor. Some need an inorganic plus an organic. Just depends on the enzyme, but whatever it needs, it's going to always need that. Once it gets everything in place to make it fully functional, we refer to it as a holoenzyme. So if the enzyme is made fully functional, you'll just call it an enzyme. But if the portion of the enzyme is not functional, the apoenzyme, once you get all of the factors together, the vocabulary term is holoenzyme. And I think of hollow like whole is what I think, the whole thing. Okay, so what else do we need to talk about with enzymes? I do want to go over naming enzymes, see if you know what some of these do. Enzymes name always end in ACE, which makes it easy. Love that. So anything that ends in ACE is an enzyme. Dehydrogenase, helicase, all of those things are enzymes. Many times lactase, many times enzymes are named after what it does or what it acts on. So lactase is an enzyme that breaks down lactose. Lactose is the substrate, lactase is the enzyme, and then that lactase acts on the substrate lactose. Dehydrogenase is an enzyme. What do you think that one does? That one actually removes hydrogens, and that one there's an enzyme that we make, humans make, produced in our liver called alcohol dehydrogenase. And what do you think that does? That is an enzyme that removes a hydrogen from alcohol and converts it into something else. So when we take in alcohol, which is a chemical, which is a toxin, which is a poison in certain dosages, when we take in, our body's job is to protect us from it. So goes into our bloodstream, circulates through, goes into the liver, comes in contact with